Hi everyone, welcome you to the GI physiology class. The gastrointestinal tract provides the body with continuous supplies of water, electrolyte, vitamins, and macronutrients.
To achieve these requires movement of food through the alimentary tract, secretion of digestive juice and digestion of food, absorption of water, electrolytes, vitamins, and digestive products, circulation of blood through the gastrointestinal tracts to carry away the absorbed substance, and finally, control of all these functions by local nervous and hormonal systems. Today, we will cover 4 topics. First, we will look at general principles of gastrointestinal motility. This picture shows the entire alimentary tract. Each part is adapted to its specific functions. Some for simple passage of food, such as esophagus. Some for temporary storage of food, such as the stomach. And others for digestion and absorption of nutrients, such as the small intestine. This picture shows a typical cross-section of the intestinal wall, including the following layers from outside surface inward. The first is serosa, followed by longitudinal smooth muscle layer, and then circular smooth muscle layer. Then submucosa, and then mucosa. In addition, mucosal muscles, sparse bundles of smooth muscle fiber, lies in the deep layers of mucosa. There are 2 types of movement in the gastrointestinal tract. One is peristalsis. It is a basic propulsive movement. A contractile ring appears around the gut and then moves forward. Stimuli that initiate peristalsis include distension of the gut, irritation of the gut epithelia, and parasympathetic nervous signals. One key feature of peristalsis in the gut is directional movement of peristaltic waves towards the anus. Peristalsis theoretically can occur in either direction from a stimulated point, but in the gut, peristalsis dies out rapidly in the all-red direction. While continuing for 5 to 10 centimeters towards the anus. This is due to mesenteric reflexes that cause relaxation of the gut several centimeters downstream towards the anus, so-called receptive relaxation. Without functional mesenteric reflexes, either due to genetic deficiency or by pharmacological intervention, directional movement is blocked. Coordination between peristalsis and downstream receptive relaxation is called a mesenteric reflex or peristaltic reflex. The peristaltic reflex plus the anal direction of movement is called law of the gut. Second type of movement is mixing movements. They are caused by either peristaltic contractions or local constrictive contractions of small segments of the gut wall. As shown in this picture, first round contractions trap bolus in smaller segments. Second round contraction break bolus into smaller pieces. Portions from two neighboring segments are mixed. Third round contraction again mix portions from two neighboring segments, as shown here. Smooth muscle in the gastrointestinal tract function as a syncytium. Syncytium refers to a single cell or cytoplasmic mass containing several nuclei, formed by fusion of cells or by division of nuclei. Smooth muscle fibers are arranged in bundles of as many as 1,000 parallel fibers. Within each bundle, muscle fibers are connected with one another through large number of gap junctions. Gap junctions allow the low resistance movement of ions from one muscle cell to the next. Therefore, electrical signals can travel readily from one fiber to the next within each bundle. The smooth muscle in the gastrointestinal tract has two types of electrical waves. One is slow waves, and the other one is spike potential. Slow waves are rhythmical changes in membrane potential caused by cyclic changes in conductance of sodium channel. For each segment of the GI tract, slow waves have a fixed frequency and variable amplitude. The frequency is determined by interstitial cells of Kayal, pacemaker cells. Except in stomach, slow waves usually do not by themselves cause muscle contraction. Instead, they mainly triggered intermittent spike potentials, which in turn excite muscle contraction. Slow waves have variable amplitude or intensity. Amplitude is affected by nervous and hormonal stimuli. Higher the amplitude, higher the spike potential frequency, which increase strength of muscle contraction. Here are slow waves, and here are spike potentials. Spike potentials are true action potentials. They occurred when the resting membrane potential become higher than -40 millivolts. They cause smooth muscle contraction. The channels responsible for the action potentials are calcium channels. Opening of calcium channels allow calcium to enter the muscle cells, which cause contraction. Frequency of spike potentials are affected by nervous hormonal stimuli. An increase in spike potential does not increase maximum frequency of muscle contraction, but it does increase the strength of muscle contraction. This information is summarized in this slide.
Now, let's move to part 2. We will look at hormonal control of gastrointestinal function.
Based on the distance that the signal travels through organism to reach the target cells, signals can be divided into endocrine signals and paracrine signals. Endocrine signals, or hormones, use the circulatory system to transport the ligands. All GI hormones are peptides. Paracrine signals, or paracrines, act on nearby cells. Some paracrines are peptides, such as somatostatin. Some are not, such as histamine. Now, let's look at the GI hormones first. GI hormones can be divided into three groups. First is gastrin family. It includes gastrin and CCK. Second is secretin family. Includes GIP, glucagon, secretin, and VIP. Third is structure unrelated hormone, Motilin. Now, let's look at the gastrin first. Gastrin is released from G cells in enterum, duodenum, and the pancreas in response to meal, especially presence of peptides, amino acids, and physical distension. In response to meal, large amount of G17 is released from enterum. G17 contains 17 amino acids. A small amount of G34 is released from duodenum. G34 contains 34 amino acids. Gastrin has several forms. G34 is longer than G17, which is longer than G14. All effects of gastrin can be produced by C-terminal 4 amino acids. Pentagastrin is a synthetic product. It contains 5 amino acids from the C-terminus of gastrin. Pentagastrin has all the same physiologic properties of natural gastrin. Gastrin increases acid secretion. It does so by two mechanisms. First, it acts directly on parietal cells that have gastrin receptor. Second, it acts on enterochromaffin-like cells, or ECL cells in the stomach, which then responds by releasing histamine. Histamine acts on parietal cells and stimulates acid secretion. Gastrin also has a trophic effect. It stimulates growth of oxyntic mucosa of the stomach, duodenal mucosa, and colon mucosa. Here is the clinical correlation. Zollinger-Ellison syndrome, or gastrinoma, is a condition with gastrin-secreting tumors. It is mostly caused by non-beta cell tumor of the pancreas, and in a smaller number of cases caused by G cell tumors in the duodenum. Excess gastrin causes hypersecretion of acid. Second effect is duodenum ulcers. To diagnose this condition, the most simple and reliable test is secretin injection. Because secretin increases release of gastrin from pancreatic tumor cells. An increase in serum gastrin is diagnostic. Second hormone is cholecystokinin, or CCK. CCK is released from eye cells in duodenum and jejunum. Stimuli are fatty acids, peptides, amino acids. CCK has several forms with different lengths. They are synthesized from the same precursor and cleaved to form different sizes. Minimum active fragment is C-terminal 7 amino acids. CCK controls secretion and motility. CCK increases pancreatic enzyme secretion. It contracts gallbladder and relaxes sphincter of Oddi. It inhibits gastric emptying. Similar to gastrin, CCK also has trophic effects. It stimulates growth of exocrine pancreas and gallbladder mucosa. Third hormone is secretin. Secretin is released from S cells of duodenum, jejunum, and ileum. Stimuli for release of secretin include acid and fatty acids in duodenum. Secretin contains 27 amino acids. Secretin inhibits acid secretion by parietal cells and gastrin release from antrum. At the same time, it stimulates pancreatic bicarbonate secretion. Because of these two functions, secretin is also called nature's antacid. It also stimulates pepsinogen secretion. Similar to CCK, secretin stimulates growth of exocrine pancreas. The fourth hormone is GIP. It stands for glucose-dependent insulinotropic peptide. It is released from duodenum and proximal jejunum. Main stimuli for release of GIP are fatty acids, amino acids, and oral glucose. Especially the hyperosmolarity of glucose in the duodenum stimulates the release of GIP. It is a member of secretin family. Major physiological effects of GIP is to stimulate insulin release. Second effect is to inhibit gastric acid secretion. That's why it is also called gastric inhibitory peptide. It is an enterogastron. The fifth hormone is Motilin. It is released from M cells of duodenum and proximal jejunum during fasting at 100-minute intervals. The release of Motilin is under neural control. It is a 22 amino acid peptide. It is unrelated to any other gastrointestinal hormones. Motilin stimulates gastric motility and intestinal motility to clear out the gut to prepare for the next meal. It increases the activity of migrating motility complex. It promotes housekeeping contraction to clear out the gut. Here is a summary of distribution of GI hormones. Most of them are produced in duodenum and jejunum. Here is a summary of stimuli and inhibition of release of GI hormones. Most of these factors stimulate release of GI hormones, except one. Acid inhibits release of gastrin. Here is a summary of main actions of GI hormones. Most of these hormones stimulate the process listed here, except three. CCK inhibits gastric emptying. Secretin and GIP inhibit acid secretion. That's why these three are enterogastron. Now, let's look at the paracrines. First one is somatostatin. Somatostatin is produced by delta cells, or D cells, of the stomach, duodenum, and pancreas. Release of somatostatin is stimulated by acid, inhibited by acetylcholine. Somatostatin inhibits release of all GI hormones. It directly inhibits parietal cell acid secretion. It mediates acid-induced inhibition of gastrin release. Histamine is produced by enterochromaffin-like cells, or ECL cells. of the stomach. Gastrin and acetylcholine increase its release from ECL cells. Histamine stimulates acid secretion from parietal cells. Histamine H2 receptor blockers such as cimetidine, ranitidine decrease acid secretion.
Now, let's move to part 3. We will look at neural control of gastrointestinal function.
Gastrointestinal function is controlled by autonomic nervous system. It can be subdivided into intrinsic control and extrinsic control. The intrinsic control is mediated by enteric nervous system, especially through two plexuses. One is enteric plexus, which is shown here. Enteric plexus lies between longitudinal smooth muscle and circular smooth muscle. The other one is submucosal plexus. It lies in the submucosa. Extrinsic control is mediated by parasympathetic and sympathetic nerve fibers. Here is the enteric nervous system. Sympathetic and parasympathetic fibers connect both the enteric and submucosal plexuses, although the enteric nervous system can function independently of these extrinsic nerves. Sensory nerve endings originated in the gastrointestinal epithelium and send afferent fibers to both plexuses of the enteric nervous system, plus to the prevertebral ganglia of the sympathetic nervous system, to the spinal cord, and in the vagus nerve all the way to the brainstem. Here's a summary of main features of enteric nervous system. It lies entirely in the wall of the gut, beginning in the esophagus and extending all the way to the anus. It is connected with extrinsic sympathetic nervous system and parasympathetic nervous system, but integration can also occur entirely within enteric nervous system and function independently of sympathetic and parasympathetic nervous systems. Some neurotransmitters are excitatory, such as acetylcholine and substance P. The others are inhibitory, such as VIP and nitric oxide. Enteric plexus controls GI motility. It increases tonic contraction of smooth muscle. It also increases contraction frequency. At the same time, it decreases sphincter tone and relaxes pyloric sphincter, ileocecal sphincter, and lower esophageal sphincter. Submucosal plexus control local blood flow and secretion. Parasympathetic supply to the gut is divided into cranium and sacral divisions. The cranium parasympathetic nerve fibers are almost entirely in the vagus nerves. They control the first half of the gut. The sacral parasympathetics originate in the second, third, and fourth sacral segments of the spinal cord and pass through the pelvic splanchnic nerves to control the second half of the gut. As shown in this picture, preganglionic neurons are long. They secrete acetylcholine. Postganglionic neurons are short. Their cell bodies are located entirely in the gut wall. They also secrete acetylcholine. Preganglionic neurons of sympathetic fibers originate in the spinal cord between segment T5 and L2. They synapse in prevertebral ganglia. They are short. They secrete acetylcholine. Postganglionic neurons are long. They secrete norepinephrine. They have their cell body in prevertebral ganglia, and they innervate the entire gut. Here are functions of parasympathetic and sympathetic nervous system in the gut. Parasympathetic stimulation in general excites enteric nervous system. In addition, 80% of vagus nerves are afferent sensory fibers. Sympathetic stimulation mostly inhibits gastrointestinal activities by either decreasing activities of enteric nervous system or inhibiting contraction of smooth muscle directly. 50% of sympathetic nerves are afferent sensory fibers. Nerve fibers secrete neurocrines or neurotransmitters. Some are peptides, such as VIP, GRP. Some are not, such as acetylcholine and norepinephrine. Most significant neurocrines are GRP. It stands for gastrin-releasing peptide. VIP, vessel-active intestinal peptide, and enteric. Here are sensory afferent nerve fibers. They can be stimulated by distension of the gut wall, irritation of gut mucosa, or presence of specific chemical stimuli. Signals transmitted through the fibers can then cause excitation or, under other conditions, inhibition of intestinal movement or secretion. Here are three types of gastrointestinal reflexes. First one is local reflexes that are integrated entirely within enteric nervous system. More specifically, afferent fibers from the gut terminate in enteric nervous system. These reflexes control secretion, peristalsis, mixing movements of the gut. Second type is reflexes that transmitted signal from the gut through afferent nerves and prevertebral sympathetic ganglia, and then back to the gut. Examples include gastrocolic reflex, in which signals from the stomach to cause evacuation of the colon. Enterogastric reflex, in which signals from colon and small intestine inhibit stomach motility and gastric secretion. Colonal ileal reflex, in which signals from the colon inhibit emptying of ileum contents into the colon. The third type is reflexes that transmitted signal from the gut through afferent sensory nerves and spinal cord or brain stem, and then back to the gut. Examples include vagovagal reflexes that control gastric motility and secretion. In this case, the signals transmitted to the brain. Defecation reflexes that produce powerful colonic, rectal, and abdominal contraction required for defecation. In this case, the signal is transmitted to spinal cord. And finally, pain reflexes. Nervous and hormonal control systems function together. Neuroactivity triggers release of hormones. On the other hand, hormones can alter neuroactivity. For example, vagus nerves secrete GRP. Gastric releasing peptide then acts on G cells to release hormone gastrin, Gastrin then acts on parietal cells to increase gastric acid secretion. This is the example of how neural activity affects hormones. Second example is CCK. The presence of fatty acids, monoacylglyceride, peptides, and amino acids triggers release of CCK from eye cells in the duodenum and jejunum. CCK acts on sensory afferent nerves, and signals are transmitted through vagus nerves to the feeding center in the brain, which inhibits appetite. This is an example of how hormones affect neural activity.
Now, let's move to part 4. We will look at gastrointestinal blood flow.
The blood vessels of the GI system are part of splanchnic circulation, in which all the blood that courses through the gut, spleen, and pancreas flowed immediately into the liver through the portal vein. Components of splanchnic circulation include gastrointestinal tract, spleen, pancreas, and liver. Arterial blood supplies to the splanchnic circulation constitutes 25 to 30% of cardiac output. Stomach and spleen are supplied by celiac artery. Pancreas, small intestine, and proximal colon are supplied by superior mesenteric artery. The rest of colon is supplied by inferior mesenteric artery. All veins from this system drain to the portal vein, and finally go to the vena cava through hepatic vein. Here is a villus. Arterial flow into the villus and venous flow out of the villus are in two opposite directions and in close proximity. Oxygen can diffuse out of the arterioles and directly diffuse into the adjacent venules without being carried in the blood to the tip of the villus. In fact, 80% of oxygen is shunted from artery to veins in the small intestine. Under normal conditions, this is not harmful. But in disease conditions in which blood flow to the gut becomes greatly reduced, such as circulatory shock, oxygen deficit in the tip of the villus becomes so great that the villus tip or entire villi suffers ischemia death and can disintegrate. As a result, absorptive capacity of the gut are diminished. Under normal conditions, blood flow in each area of the gastrointestinal tract is proportional to local activity. After a meal, for example, blood flow to the gastrointestinal tract can increase 2 to 3-fold for 3 to 6 hours. Causes of activity-induced blood flow include hormones released during digestive processes that promote vasodilation, such as gastrin secreting CCK. Two kinins released, calidin and bradykinin. They are potent vasodilator. Low oxygen level or high adenosine level can increase intestinal blood flow 50 to 100%. In addition, parasympathetic stimulation increases activity of stomach and lower colon, and secondarily increase blood flow in this area. Conversely, sympathetic stimulation causes vasoconstriction of the arterioles and decrease blood flow directly, such as during exercise and shock. However, After a few minutes of this vasoconstriction, blood flow often returns to normal. The local metabolic vasodilators that are generated by ischemia override the sympathetic vasoconstriction, returning toward normal the necessary blood flow. This phenomenon is called autoregulatory escape.